Hello lovelies, in this video we're going to be looking at the chloroplast, its structure and its functions for your A-level biology. Once you've finished making loads of notes for this, you're going to come across lots of new words which you need to properly define in your notes. That's really important that you do that. You can pop over to the website and try all the free questions on there to help you with your recall.
Hi everyone. Okay, so we're going to start looking at the chloroplast structure and its adaptations. So we're going to have a look at the structure of the chloroplasts, all the label structures you can see here, explain what they do, and then link them to their adaptation for photosynthesis. Then we're going to have a look at the photosynthetic pigments that you can find in the chloroplasts and how they help to absorb light for photosynthesis and how they're arranged in their structures. And then we're going to have a look at how you can extract those pigments using chromatography, which is one of the required practicals. So let's have a look at the chloroplast structure. Much like with the cell and the cytoplasm, the stroma is a gel-like fluid that is inside the chloroplast, and it contains all the things that are required for reactions to happen in the stroma. So it's got enzymes, it's got proteins, and it contains some other things that are useful to the chloroplast and allow it to carry out its functions. So there are starch granules, which are obviously used as a method of storing glucose, which we're obviously going to be producing from photosynthesis. There's also DNA and ribosomes, which allows the chloroplast to make its own proteins that it needs for photosynthesis, which means it doesn't need to rely on them coming in from the outside cell. The really interesting thing about the DNA and the ribosomes and their presence is that they're evidence that the chloroplasts were probably once free living bacteria. The chloroplast DNA is circular, much like the bacterial DNA. The ribosomes are 70S ribosomes, which are also clearly the same as bacterial ribosomes, the smaller kind. And so there's evidence here that at one point chloroplasts were able to be living free like bacteria did, and then at some point they would have been engulfed or absorbed into another cell, which is how they then became part of what we now have as eukaryotic cells. And this theory is known as the endosymbiotic theory. The double membrane that you can see around the outside is also evidence for that fact. So at one point they would have been engulfed into a cell in a vesicle, and that's what created that double membrane. So we have an inner membrane, the outer membrane, and then the space between those two, which is just known as the intermembrane space. These are important, and again, much like the cell membrane, it works to control what substances come in and out of the organelle. The outer membrane is permeable. It's permeable to gases and permeable to water and all the other things that we need to get into the chloroplast to do photosynthesis, so that's very important. The inner membrane is slightly less permeable, so it relies a lot more on transport proteins and allows the chloroplast to control more what comes in and out of the chloroplast, which is obviously really useful and can control things like the reactants and products we need, and uses transport proteins to control the different amounts. One thing we talked about when you would have done cells would have been compartmentalization. So the chloroplast is very much, the inside and the stroma inside is very separate to anything that's inside the cell. It can have a very different makeup of concentrations and enzymes. Then what we're going to look at lastly is the other type of membrane. There's actually a third type of membrane. So there's the inner membrane, the outer membrane, and then this third type of membrane is actually the construction of these disc shapes that you can see inside the chloroplast. Much like you have the folds, the cristae, the folds inside a mitochondria, we have our own membrane inside the chloroplast that's folded in or structured in a specific way. It's known as the thylakoid membrane. All of the structures you see inside are made of this thylakoid membrane, and the membrane itself is what contains the photosynthetic pigments that do the absorption of the light. So the thylakoid or athylakoid is a disc or a sac of this membrane, and it contains, like I said, it's made of membrane, so it has the photosynthetic pigments in the membrane that are going to absorb the light, and it has like a thylakoid space or lumen inside that disc. These thylakoids are stacked up together on top of each other like a stack of coins, and one stack of thylakoid discs is known as a granum, and then there are lots of them you can see. So between these stacks of thylakoid discs, these granum, is the lamellae. And that's just sections of membrane, strips of membrane that join those granum together. They tend to dictate the distance between them, hold them apart. And again, they're made of thylakoid membrane, same, and they contain the pigments as well. Multiple granum, it's the singular, are known as grana. Lots of grana are the discs, the stacks of discs inside. So this should hopefully let you recognise what chloroplast looks like. This will be a recognisable picture for those of you who've seen it inside cells, and hopefully you'll be able to recognise some of these structures in electron micrographs as well. Here we've got the first kind of look at how these structures are adapted for photosynthesis. So the stroma, like we said, has got enzymes in it, and these are important because the stroma is where the light-independent reactions happen. So the reactions in photosynthesis that happen without light. We'll come to that in the next video. Enzyme reactions have to happen there, and so they've got enzymes ready to do that. The one main one is ATP synthase. We're going to need ATP to do photosynthesis. The other thing is that within the stroma we've got lots of proteins that we need, we've got lipids that we need, and obviously we're storing starch as well, which is obviously the way we're glucose, which is being produced by photosynthesis. So we've got the outer membrane, so the inner and the outer membrane, which helps the chloroplast to control what's coming in and out of the organelle, which is important for photosynthesis to make sure we get in enough of what we need, and also to control how much is leaving and the substances that are leaving them through the membrane. It also is permeable, especially the outer membrane, which allows our gases to diffuse in and out that we need for photosynthesis to take place. Because chloroplasts have their own DNA and their own ribosomes, they can do their own protein synthesis, which means they can make their own proteins that they need for photosynthesis within the chloroplast itself. They're not relying on the cell, which helps speed up the process. And lastly, you've got the thylakoid membrane. So there's a large surface area for light absorption, which means there's more pigment. So the way it's arranged, the way we're stacked in these grana with these thylakoid discs, the more surface area membrane we have, which has the pigments on, which means we will be able to absorb more light, and that means more photosynthesis. Also, there's a large surface area for enzymes. So again, ATP synthase is one of those as an example that we need, and the proteins embedded in those thylakoid membranes that we need for the light-dependent reactions. That's where the light-dependent reactions occur. They occur on the thylakoid membrane. So the more membrane we have and the more surface area we have to do that, the more light-dependent reactions are going to happen. And also the grana, the stacks and the thylakoid membrane, there's a lot of those edges exposed to the stroma. So that means that a lot of all the products from the light-dependent reactions can go to the light-independent reactions quite easily, and they can exchange products and reactants because they're close by each other because the stroma is constantly surrounding and touching the thylakoid membrane. So all of these make our chloroplasts really well adapted to carrying out photosynthesis. We're going to have a look at now photosystems and the pigments inside them. So you can see here we've got a picture of a photosystem. A photosystem is this area here. So it's this green section here. And you'll notice that it's in the membrane, so it's embedded in the thylakoid membrane because on one side we have the stroma and on one side we have the thylakoid lumen. So if you can imagine, we've taken this photosystem from the edge of the thylakoid membrane. So this is one thylakoid disc, and this is where our photosystem is positioned inside. So a photosystem, we've cut through its cross section. So if you had this as a whole object, it would be funnel shaped. So the idea is it funneling down to a point, and it's a protein complex. So it's multiple proteins joined together within this membrane. The whole point of a photosystem and its aim is to harvest light energy. So you'll see here, light is coming down and hitting the photosystem. Its job is to take light energy and use it to excite these electrons here in the chlorophyll in the center, and we're going to raise them to a higher energy level. So we're taking the energy from the light and we're using it to excite an electron. And ultimately photosynthesis is doing that. This is the first stage, and it's going to turn that energy from the light that excited that electron into chemical energy and building those bonds with glucose. It's going to take some pigment to absorb the light energy. So we have chlorophyll here in the center, and we also have some other pigment molecules here. But our chlorophyll is the aim, is where we're going to excite the most electrons. If you look, the chlorophyll is in the section that we call the reaction center. Okay, so the reaction center is where we have our chlorophyll, and that's where the exciting of the electrons is going to be happening in the reaction center. Now, the chlorophyll at the base of our reaction center in all the photosystems is chlorophyll A. There are two types of chlorophyll, but they both have in common the fact they have a magnesium ion at the heart of the molecule. That is what allows the chlorophyll to do its job and get those electrons excited. And so without magnesium, the plant cannot make chlorophyll. So that's why it's an essential nutrient that we have to actively transport in through the roots to make sure that we have enough magnesium to make enough chlorophyll, to have enough chloroplasts to absorb for photosynthesis. Chlorophyll B is another type of chlorophyll. It's mostly used as an accessory pigment, which are the pigment molecules you can see here in the sides of the photosystem. We'll come to those in a minute. So the light comes in, as you can see, and it hits these pigments in the edge of the photosystem. Now, what you can kind of see here is this light energy being passed between those pigments to the chlorophyll molecule at the bottom. So these accessory pigments are really important because they are the ones that are trapping the light energy from photons, which are the energy carrying molecules in light. And the energy is being passed between them in order to reach the chlorophyll at the center of the reaction center. Now, there are actually two types of photosystem. There's photosystem 2 and photosystem 1, and I'm saying them that way around because often you'll see that they're in that order because that's the order that light hits them in the process of the light-dependent reactions. They go backwards 2 and then 1 because they were discovered first. Photosystem 2 was discovered second, photosystem 1 was discovered first, but they actually go the other way around. And so they both have chlorophyll A at the bottom, but one of them, the chlorophyll A, absorbs light at 680 nanometers, and the other one, chlorophyll absorbs light at 700 nanometers. That's their kind of peak absorption wavelength of light that they are absorbing. And we'll talk about them in the next video, about the light-dependent reactions, about how they work and how they function in the light-dependent reactions, but you just need to know that there's 2 of each. Photosystem 1 is found most commonly on the lamellae, and photosystem 2 is found most commonly on the granum. And as I said, the 700 and 680 are the wavelengths of light that they absorb the best. Most plants actually have more pigments in them than just chlorophyll. So they have many photosynthetic pigments within the photosystems, within the thylakoid membrane in their chloroplast. And that's because that increases the range of wavelengths absorbed and therefore increases how much photosynthesis they can carry out. These accessory pigments are what we call them and listed in the table. And as we said, they are part of the photosystem that helps here to channel light down into the chlorophyll. And as you can see, they appear different colours because they have the ability to absorb different wavelengths of light and therefore they reflect other colours of light. So you'll notice that chlorophyll A obviously is on the list, but that's not an accessory pigment. The others are all the accessory pigments that you can find in the thylakoid membrane in photosystems. And if you add all of these up together, you'll be able to see that the range, so from 375 all the way up to 700 if we include chlorophyll, but even that if we go up to 640 with chlorophyll B, we've got a larger range with them all added together for all the different wavelengths of light that can be absorbed than if we just had chlorophyll on its own, because there are different pigments like the xanthophylls and the anthocyanins can go into the sort of more blue purple areas and a bit into the green, and then we have obviously most of the light being absorbed being in the red end of the spectrum and the blue end of the spectrum. As you can see by this graph here, the photosynthetic rate versus the colour of the light, this is what we call an action spectra. So it's showing you the colour of light, the wavelength of light, and how that affects the rate of photosynthesis. So as you can see, the more blue light, the more the sort of 450 nanometer light, the more photosynthesis, and then again we have another peak in the red end of the spectrum. So this is showing you how the wavelength of light can actually affect the rate of photosynthesis. There is another type of spectrum you can see which just shows you the absorbance of each type of pigment at each wavelength. The other type of graph is called an absorption spectrum. So it would look like this. So you'd have your absorbance on the y-axis instead and a wavelength on the x-axis, and then you can get different graphs that show just the absorbance of chlorophyll at a specific wavelength. So a peak shows that it absorbs the most light at those wavelengths. So normally you can lay one of these on top of an action spectrum and it will show you that the more absorbance you get, obviously the more light being absorbed, the higher the rate of photosynthesis, which makes sense. There will be plants that are adapted to shade or adapted to UV, which have different ratios and different numbers of each of these pigments in their leaves. So UV adapted plants, so they have light to protect them from UV, often have lots of xanthophylls, for example. So they will absorb the harmful UV rays. And also we have shade tolerant plants. They're not absorbing the full spectrum of light, because they're growing underneath other plants, they're growing in shade, so their light is not going to be the full spectrum, so they don't have to choose. It's going to be a lot of light that is being passed through other plants. So they will have a different makeup of pigments to be able to be adapted to absorb the kind of wavelengths that come through being reflected through or passed through other leaves. Okay, so the required practical 7 is chromatography, and the reason it's here and in this part of the spec is because we're looking at being able to show and demonstrate that plants contain all of those different accessory pigments that we've talked about, and then to be able to do it for different types of plants, so shade tolerant plants or UV resistant plants, in order to show the differences by identifying those pigments using something we call the RF value. Our method is on the right hand side, so let's go through the method and talk about why the justification for those steps, and then we'll look at how we calculate the RF value. We're going to start by grinding up some leaf tissue. Obviously we're going to put a bit of sand in to act as like grit tools to try and make it grind, and you're going to use a pestle and mortar. This is to break open the cells because we need to get the chloroplast out of the cells, and then hopefully try and destroy the chloroplast a little bit as well to get those pigments and those membranes out. We're going to add a solvent and continue to grind it up. Think about when you did factors affecting membranes permeability. So it's going to disrupt and destroy the membrane, get those pigments out, those photosystems out, and dissolve it in that solvent. Then we're going to draw our pencil line, okay, which is going to be the start position. We're using pencil, not pen, because it won't dissolve in the solvent, so it won't run up the chromatography paper along with your pigments. Then you're going to make a small spot from a quantity of your leaf extract, which should be kind of liquidy now, and you do a small spot, you leave it to dry, small spot, leave it to dry. We normally use a capillary tube. This is to try and concentrate that leaf extract on that spot, so you get a lot of pigment in there. Dip the bottom of your chromatography plate or paper into the solvent and suspend it, normally with some kind of bar or a bung or something to hold it up. You're not going to put the solvent high up enough to cover the spot. You're just going to touch the end of it. We don't want to dissolve the spot in that liquid at the bottom. We want the spot to stay where it is and the pigments and things to just be carried in the dissolved solvent, moves up the plate through absorption. It can be hazardous. The propanone and other solvents you're using can be hazardous, so you should be doing it in a fume. It should have a lid on it. It shouldn't be exposed or emptied out for long periods of time. Also, it can evaporate, and we don't want all our solvent to evaporate out of the jar. Then you're going to leave it to run up, but just before the solvent reaches the end of the paper, when you can still see a gap, you're going to draw a pencil line across the top. Again, because once you take it out and you leave it to dry, that solvent's going to evaporate and you're not going to be able to see where it travelled, and we need to know the distance the solvent travelled to calculate our RF value. I would suggest taking a photograph like I have of the one over here on the side that you can see from my students. This is a photograph I took because the colours will fade, the spots will fade because they react with light. So the faster you can get that done, so drawing the solvent front here, which is this top line, so drawing this solvent front in pencil, putting maybe a dot in the centre of each of the pigments, taking a photo, noting down the colours that you see before they fade as well, so that you can help start to try and identify those pigments. So then the last thing we're going to do is calculate the RF values. So calculating the RF values of each pigment will allow us to hopefully try and identify what pigments we've got. The thing you have to remember is that they are unique to the running solvent. So the RF values we have calculated will be specific to the running solvent we use. If we use propanone, then these will be the values for our leaf tissue for propanone. They won't be able to be compared to other solvents because it will be different each time. This is because although the pigments are separated out by mass, they're also separated out by their interaction with the solid plate or the paper, and also how well they dissolve in the solvent dictates how far they move. So you are not going to be able to get the same result necessarily, the distance moved in the same way with different solvents, because they're going to interact with the solvent in different ways. Now we've got all of that, you can see from my example over here, we've got this as our final product. We've now got 1, 2, 3, 4, 5 different pigment spots, and this is our original spot down here. We want to be able to measure the distance from the start line to each pigment spot to know the distance that it travelled, as well as the total distance travelled by our solution, which will be from the start line here to the solvent front here. So this is our distance travelled of our solvent. And once we've got those two numbers, we can calculate our RF values for each pigment using this formula. So once you've got that, you've got your RF value, you have the colour, and you know how far up it moves. So our yellow pigment here ran really, really far. versus sort of the orangey one here. And you've got the colours, so things like the grey, we know that grey might well be PhET because it is identified as grey, but we would have to check on various tables. But once you've got that, you can repeat the process again with a different type of plant, a shady plant versus a sunny plant, or a UV protective plant versus another one, and just see what the differences are, compare them, look at the differences and see what different pigments those plants have and try and link those to their adaptations. So to sum up, we looked at the adaptations of chloroplast. So the stroma is where the light-independent reactions happen and it contains the enzymes, including ATP synthase. The thylakoid membrane is where the light-dependent reactions happen. It's where the light is being absorbed. So the stacking of those grana and those thylakoid discs creates a large surface area for light absorption and a large surface area for the enzymes and the proteins that are within the membrane to help carry out the light-dependent reactions, which means more photosynthesis is going to happen. You've got the permeable membranes around the outside that allow the gases to enter and exit for photosynthesis and also have that level of control. We've got the accessory pigments in the photosystems that increase the wavelengths of light that can be absorbed, and so more photosynthesis takes place. And then we looked at how you can show that those accessory pigments are there by using the chromatography method from Repair Factor 7 to separate and identify those pigments from different plants and maybe compare them.
Ouch. That'd be too grim.